One thing scientists say is very clear. Using gene editing on humans is still a long way off because we do not know what happens when an embryo with edited genes actually grows into a person.
These are some big unknowns that it'll take years to understand, and we're not really ready for this to get launched. The ethics, the unknowns are really big questions.
After the failure of similar attempts in China, scientists at Oregon Health and Science University used a different approach, trying to repair a faulty gene in a human embryo at the moment of fertilization. Sperm from a male donor that scientists knew were carrying a mutated gene which causes a hereditary heart disease were inserted into human egg cells together with a molecule that serves as genetic scissors directed at the faulty gene in the male DNA.
So DNA is a kind of long string, and these molecular scissors would cut where the mutation is.
Out of 58 fertilized eggs, the cut was repaired in 42 of them that continued to grow without the faulty gene. The experiments were stopped when the embryos grew to only 8 cells.
If you look at the 8 cells, every cell would have perfectly the same repair.
The experiment showed that if gene editing is done at an early stage, the cells can repair the cuts and continue to grow without disease-carrying genes that have plagued families for generations. But it is not yet known whether the embryos with edited DNA would grow into healthy humans. Current U.S. laws do not allow clinical experiments on humans, but other countries have less stringent regulations. George Putic, VOA News, Washington.